I just first of all would love to hear if you guys can hear me okay, because this is my first live stream. So let me know in the comments if this is loud enough and if you can hear me loud and clear. Zishan, I see you've said, hey, so that's really good. Did you manage to hear this response okay? Okay, so I'm just going to be posting a few comments. I think everybody will be able to see these when they join. I've had quite a few people asking me questions on Instagram. So I'm thinking there's hopefully going to be a few more people joining us at some point. So yeah, do bear with me. This is my first live stream, so if there are any issues, I'll try and resolve them as quickly as possible. Okay, hey Alicia, we've got a few more people joining. Let me just get the rest of my message up, and then we're going to get started with me working through your questions. Okay, so I'm guessing most of you have joined me from Instagram. This is going to be a live stream for paper 1 of biology. I'm going to be working through AQA specification. That's what I teach. So hopefully I can help you with that. If you are on a different exam board, this will still be relevant for most of the topics, so you can still join in. I'm going to cover both foundation tier and higher tier. If you're doing combined science, I'm covering that too. And I'm also going to cover biology-only topics if those questions come up. Alicia, I can see that you're saying you do OCR. So I haven't ever taught OCR. I've taught AQA and Edexcel. So I'm afraid I won't know exactly how much overlaps with that exam board. You can listen in, and if it doesn't, then you can just tune out for those bits. Okay, so can we start with some combined higher AQA, please? Yes, of course we can. So I've had a few, I've made a list. I've had a few topics sent through on Instagram. So I'm going to start by working through those topics. One that has come up twice in my requests is mitosis from GCSE Saver. They just want to run through mitosis, and another person has asked for the difference between meiosis and mitosis. Just to quickly address that, you shouldn't get a question that asks you to compare meiosis and mitosis on paper 1 because meiosis is on paper 2. So that question shouldn't come up until paper 2. I'll cover it briefly just because you've asked, but you don't need to worry about it for tomorrow's exam. Okay, so first up, there is a cell cycle. Cells aren't constantly undergoing mitosis unless they're cancerous cells. So the first part is when, and this is the biggest part of the cell cycle, is that the cell needs to replicate its DNA, and it will also replicate the organelles that are inside it. So that's things like the mitochondria and the ribosomes. And then once that's done, the cell is ready to enter mitosis. So mitosis is the actual division of the nucleus. And essentially what happens is the chromosomes, which is what makes up your DNA, those have doubled. So let's say we start with 2, you will now have a cell that's got 4 chromosomes. The chromosomes will split and move to opposite ends of the cells. So you'll have 2 on one side and 2 like that on the other side. And then the cell just splits in half. And that splitting of the half is the third stage of the cell cycle. So, I mean, there's not a lot more to mitosis than that. Your teachers may have taught you more detailed steps, which aren't needed for GCSE, because there is a lot more detail that you can discover if you go on and study A-level biology. But the simple steps are the DNA doubles, and so the chromosome number doubles. then the chromosomes move to the opposite ends of the cell, which you can also call the poles of the cell. And then the cell divides into 2 daughter cells. And that's it. That is all you need to know for mitosis. Let's address the difference with meiosis then. The nice thing is that meiosis starts off exactly the same. It starts off with one parent cell, doubling the DNA, that number of chromosomes splitting in half, but then the cells undergo another division. So there's a second division in meiosis, and that then means that there is half the amount of DNA in each of the daughter cells as there was compared to the parent cell. So if you started off with, again, 2 chromosomes in your parent cell, those then double, so you get 4. They split in half for the first division, so there's now 2 in each cell, and then they split in half once more. So you end up with 4 daughter cells, each with 1 chromosome, which is half as much DNA as the parent cell. And in meiosis, that happens to make gametes. If you've studied paper 2 or revised it yet, hopefully you know that gametes are the sex cells, which are the egg and the sperm in humans, and which are the egg and pollen in flowering plants. So meiosis is for gametes. Mitosis is just for any other cell in the body. So that's mitosis. Have you got any questions in the comments about mitosis? Is that all clear? If it is, we will move on. Okay, so another question I've got from Instagram is from Billy. Hey, Billy, if you're there. Billy wants to know about food tests. So this is one of your required practicals that you need to know, and it's in the topic of organization and digestion. Okay, so there are 4 different food tests that you need to know. The first is the simple sugars, or just glucose. So that's the common one that you learn about. So you test for sugars using Benedict's. And Benedict's is this nice bright blue solution. And so you'll add your simple sugar, your glucose, or whatever food it is you're testing for glucose, to your blue Benedict's, and then you heat it up, and it should go red if there is glucose present. It's really important with food tests that you say the before color as well as the after color. Okay, so make sure you put down both colors in your answer. So Benedict's for sugars will turn from blue to red when heated. Okay, next up are your complex sugars, or more simply, carbohydrates like starch and glucose. So this test is iodine. You'll have used it, I'm sure, 100 times in your science lessons, that classic leaf test for starch. So iodine, this is important actually. People do lose marks here because they don't use the exam board colors. They just make up colors. The way that AQA describes iodine is as yellow-red. So that's the color of it at the start. It's yellow-red. And then if starch is present, the iodine will turn blue-black. It's a bit annoying because you actually have to say 2 colors, yellow-red to blue-black. But that's really important, so make sure you put down those exact colors. Yellow-red to blue-black if starch is present. Okay, the third food test you need to know is for lipids. Lipids are generally fatty foods like olive oil, butter, etc. So this is the ethanol test, and all you do is mix your food with ethanol, and if a white cloudy layer forms, then lipids are present. So that's just a cloudy white layer. Ethanol, it's a type of alcohol, so it's just colorless. AQA actually don't put that in the specification, so I don't think you need to have the before color in this one. But just to be on the safe side, I'm telling you that it's colorless, and it will have a white cloudy layer if lipids are present. There's a little bit of health and safety for that one you need to know. Ethanol is harmful, which means you shouldn't obviously ingest it or drink it, take it into your body. It's also flammable, so no naked flames should be around it. I've seen Zane has said, can we use the Sudan 3 for lipids? I would question what exam board you're on, Zane. If it's AQA, you need to say ethanol, That is another test. That's red, isn't it? That chemical. But for AQA, scrap saying Sudan 3, say ethanol. That's what's in the specification. So say that. Okay, and then, Lauren said something helpful there about saying iodine solution. That's interesting. So I have got the specifications up in front of me. Hopefully you can still see me while I'm searching. So I'm going to just search very quickly and see what it says. Iodine test for starch, which doesn't make it sound like you have to say solution. Let's go a little bit further back here. Yeah, I'm not sure. Well, to be on the safe side, why not say iodine solution? Okay. Oh, and finally, the last test is for proteins. That is the Burette solution, or the Burette test. And that's spelled B-I-U-R-E-T, just to check your spelling there. And that is blue to purple if proteins are present. So that's your last one, blue to purple, a nice easy one. There is some health and safety with the Burette test as well. This solution is corrosive, which means you shouldn't get it on your skin. You should be using eyewear to protect your eyes, so safety goggles. And that's the end of the food tests. Okay, any more questions with that? No, hopefully all good. Okay, so let's see. A few people have asked, can I go through questions that are likely to come up tomorrow? It's really risky territory for me to make predictions. And I think it's safe to say for any teacher or tutor, etc., to make predictions. The reason being, there have been so few tests on this. Last year was the first new specification exam, which means we have nothing to base it on. And a lot of people might think just because something came up last year means it's not going to come up this year. Well, that's absolutely not the case, and you should be really careful if you've been told that. Things can come up in repetitive years. So to be honest, I think it's far too early to say. Maybe if it was a few years down the line and there was an obvious topic that hadn't been covered, then I would be betting for that. But I think at this point, absolutely anything could come up. So I'm sorry, I can't give you an easy answer there. I couldn't confidently make any predictions. Okay, so I'm going to finish up with what else was on my list. It's quite short now, so that's good, so I can get to some of the questions. So from Instagram, I had a request asking how antibodies work in protecting you during the immune response. There's actually not much you need to say here. You'll learn loads and loads about antibodies and the follow-on response from that if you do A-level biology. But on the GCSE specification, it's really short and sweet. All you need to say is that antibodies are specific to the bacteria's antigen. And I'll explain what an antigen is. That is just a protein that is on the surface of a bacterium or a pathogen. And it identifies the bacterium as foreign. So when your body encounters a bacterium or pathogen, it's the antigen that tells it that that's a foreign body, it shouldn't be there, and that's what it needs to respond to. Okay, so antibodies, they're specific to the bacteria's pathogen, the bacteria's antigen, and they attach to it, causing the bacterium to be destroyed. That's it. You don't need to know how they cause that destruction. You just need to know that they can attach to each other and cause the destruction of the bacterium. Okay, so the last thing was required practicals. And I think I saw a question towards the top saying, could we go through required practicals? Is that still there? Maybe I can't see it anymore. Truthfully, there are obviously a lot of required practicals. To go through every single one would more than exceed the time we have here. If you've got any specific questions about a required practical, please add those now and I'll take a look at those. Okay, Erin would like to go through chapter 9, respiration. So respiration is quite a straightforward topic on the surface, but you're going to get loads of data questions here. To start with, the important things are that you know the difference between aerobic and anaerobic respiration. So aerobic respiration uses oxygen, it combines with glucose and will produce carbon dioxide and water. I'm sure you know, but it's just the reverse of the photosynthesis equation. So it's just the same equation flipped. So that's aerobic respiration. Anaerobic respiration is with lack of oxygen or absence of oxygen. So it's just glucose followed by your arrow, lactic acid. That's in humans for anaerobic respiration, but it does also take place in yeast. And whenever it takes place in yeast, instead of lactic acid, it produces ethanol, which we've mentioned before is a type of alcohol, which is toxic. And then it also produces a small amount of carbon dioxide in yeast. So the differences between aerobic and anaerobic respiration, the main one is that aerobic respiration produces much more energy. And I've just used a word you're not allowed to use. It releases much more energy. Really important to highlight, you can't say the words create, produce, or make energy. Those will lose you a mark, especially when you get a question like describe the function of the mitochondria. If you say it makes energy, you're getting no marks. You have to say it releases energy through the process of aerobic respiration. Okay, so releases is the buzzword here. Okay, so aerobic respiration releases much more energy than anaerobic respiration. And this is because it is a more complete breakdown of the glucose with the oxygen. Anaerobic respiration will cause fatigue because lactic acid is building up in the muscles, and that needs to be removed. There's a little bit more detail on that. Maybe, Erin, I'll ask you, are there any specific parts of the respiration topic that you want to go through? It is, like I said, a fairly chunky one, but actually fairly data-based. So you're going to get a lot of graphs with this sort of one. Okay, let's take a look at some more questions. So apparently there's likely to be a 6 marker on diffusion. Maybe there will be, yeah, more than possible. Can we go through bioenergetics? Well, that's a little bit of it covered. Respiration. Would you like to go through some photosynthesis, Zane? Would that be helpful as well? Okay, so I'll briefly go through photosynthesis since we've got a bioenergetics request. Like I said, photosynthesis is the reverse of respiration or aerobic respiration. So photosynthesis is carbon dioxide plus water, and the carbon dioxide comes from the air, which is taken in through the stomata in the leaves, which are just the little tiny holes in the underside of the leaf. So carbon dioxide plus water. Where does the water come from? Yep, from the roots. That's absorbed by osmosis, which we can cover if you need more detail on that. And then arrow, you will produce glucose. That happens in the leaves because that's where photosynthesis happens, the leaves and stem, the green parts of the plant. And then oxygen is released, and again, that can move out through the stomata in that process of diffusion, or it might actually get used inside the plant for respiration, because remember, all living things carry out respiration, including plants. So plants do use up some oxygen. Okay, so that's photosynthesis. The products of photosynthesis we'll talk about briefly. So I mentioned oxygen obviously can pass out of the stomata or can be used in respiration, but glucose is the food of the plant. So this will need to be transported in the plant, and you need to know how that happens, just briefly. So do you know which organ is an organ of the plant transports glucose? See if you can comment in the chat. Which plant organ transports glucose? First person to answer gets a shout out. Hey, well done, Zane. You're first. So it's the phloem. I think there might be a slight lag. Apologies about that. Okay, so the phloem transports glucose. It is mentioned in the specification that you need to know the adaptations of the phloem. So there's just really two. It's an elongated tube, which means it's just stretched out. So it's nice and long in the plant. And these are living cells. And at the end of it, they've got pores, just like the pores in your skin. It's tiny holes, and that allows the cell sap, which is essentially the glucose, to get transported through the plant. It needs to be transported because the roots cannot make glucose. They don't have any chloroplasts in their cells, but they still need to respire to release energy for survival and for active transport. So the glucose or sugars need to get down to the xylem, to the roots. I'll also briefly mention the xylem. So the xylem is for transporting both water and ions. So those are taken in through the roots. I'll ask another question for the comments. Please have a go at answering. It's quite hidden in the specification, but it is in there. Do you know which material is used to strengthen the xylem cells? Xylem cells are dead cells. They're hollow, but they are strengthened by a material. Well done, Zane. You're on the ball. Laura, Erin, wow, you all got it. Okay, and Hannah too. Okay, so lignin is used to strengthen the xylem cell walls. Okay, so that's water and ions are transported through the xylem up from the roots in the transpiration stream. As we mentioned, the stomata are open, which does also allow water to be lost from the leaves. So it's important that that water loss through evaporation is replaced. So we need to transport it up the plant through the xylem. Okay, so that's a lot covered from bioenergetics on photosynthesis. This is relevant, Laura, so yeah, I'll go through what guard cells do. Okay, so guard cells, they are either side of a stomata. So a stomata is just a hole. It's not actually a physical structure, it's just like the hole. My hands right now are the guard cells. So they are able to open and close this hole. So at the moment, our stomata is open. The guard cells can they can rearrange their shape to close the stomata. And you might wonder why do we need to close the stomata? So there are certain conditions that can cause water loss to really speed up in a plant. It's not good for plants to lose too much water. A, because they need it for photosynthesis, and B, because they will wilt and die. So a plant needs to conserve water loss. So the conditions that speed up water loss, or transpiration is the word we should use, are things like it being dry, it being windy, it being hot, and initially having a high light intensity, because that means lots of photosynthesis is happening, so the stomata will be open to allow gas exchange. But if it gets too high and too much water is being lost, then the guard cells need to close to stop water loss or evaporation of water through the stomata. So it's not explicitly on the specification, but you could get an application question that will require you to explain how that works. The guard cells change their shape by having water move into them. So when water moves into a plant cell, it becomes turgid, which just means it is like full. It's like completely filled up with water and it's big. So the movement of water by osmosis is what fills up your guard cell and causes it to close. Hope that helps a little bit. Okay. Can I go through the heart, and can we go through diffusion in animals and plants, and how do we find limiting factors on a graph? Okay, that's a lot. And what practicals are there? I'll go through the practicals actually, just since we've already mentioned it very quickly. So I've jotted them down. Here you go. We've got microscopes, comes up. Then we've got, I don't know if is this backwards for you? Maybe. We've got a biology only topic, which is the use of antibiotics and antiseptics to see their effect on bacterial growth. We've got the osmosis practical, which is using different concentrations of salt and sugar solutions on plant tissue. And then we've got food tests, which we've been through on here. And then we've got the effect of pH on the rate of amylase reaction. So those are the required practicals. I would really recommend that you use your specification, though, to double check which ones you need to know. There's no There's no difference between the higher and foundation required practicals, but there is a big difference between combined science required practicals and biology only required practicals. So just check your specification for that. Okay, so Hania wants to know about the heart. I really like the heart. It's a nice, fun, visual topic. I might do a little drawing. Can you tell me, first of all, is this back to front for you? When I look at the screen, it is. It's not back to front? Brilliant. Okay, so I'm going to draw a little diagram of the heart, which will speed things up a bit on that. So first of all, the heart is flipped. So when you're looking at a picture of it, we call the right side the left side. We call the left side the right side, just to make it nice and confusing. There are two tubes entering the heart, and then there are two tubes leaving the heart. And then we've got 4 chambers in the heart. Okay, so you need to know the names of the 4 chambers. And then you need to know what type of blood is coming in each, the names of them, etc. Okay, so here's my rudimentary diagram of the heart. So we'll start with, I always start just over here with the blood coming back from the body. So when it comes back from the body, the blood has got a low concentration of oxygen. It's not got no oxygen. It still has got some. Your cells don't use up 100% of the oxygen you breathe in, but it is lower. So you can call it deoxygenated blood. And that comes in through this tube, which is called the vena cava. Lauren, seeing a quick question there. You don't need to know bicuspid or tricuspid for biology, just for PE. Okay, so the vena cava is a vein that brings deoxygenated blood back to the heart. I like to remember that it is V for vein. It then brings it into the right atrium, because remember, it's back to front. So even though this is on the left side of what we're looking at, it's flipped, so it's the right atrium. It then goes down to the right ventricle. Here's my hint for remembering which way is which. I think the shape of the heart, hopefully they give you a good diagram in the exam, I think it looks a bit more like a V, because it kind of comes into a bit of a point at the bottom of the heart. So the ventricles, they're the ones on the bottom. So let me continue my little arrows. Right atrium to right ventricle. It then leaves the heart and it will go to the lungs via the pulmonary artery. So arteries always carry blood away from the heart. So the A is for away. That's how I remember what arteries do. Arteries or away from the heart. So this pulmonary artery is actually the only artery in your whole body that will carry deoxygenated blood. All of the other arteries carry oxygenated blood. Okay, so the pulmonary artery is going to take that deoxygenated blood, that gets a little pump to take it up to the lungs. So in the lungs, that's obviously where you've breathed in a new breath of breath of air, and you're going to do gas exchange. So the carbon dioxide that's built up in this blood is going to get removed in the lungs, and oxygen is going to enter it instead. And those both happen through diffusion. So it picks up the oxygen in the lungs, and then it's going to come back to the heart. And because it's coming back from the lungs, we still call it pulmonary, but this time it's a vein because it's coming into the heart. So the pulmonary vein will take the blood that's now oxygenated into the left side of the heart. And remember, it's atrium at the top, so it'll take it from the lungs through the pulmonary vein into the left atrium. And then this one looks a bit more like a V here, is the left ventricle. And then in the comments, see if you can tell me what we call this main artery that leaves the heart with the oxygenated blood. Do you know what this one should be labeled as? This is the main artery that's going to take all of your oxygenated blood to the body. Yeah, good. We've got lots of answers. It's the aorta. Very good. Okay, so that's it. There's a little bit in the specification about the heart that mentions that our human cardiovascular system is a double pump, and there's an important reason for this is because there are kind of two parts to it. So one part is the left side of the heart is to do with your majority part of your body, and the right side of the heart is to do with the lungs. So obviously you've got a huge network of capillaries all over your body, as well as your arteries and your veins, and diffusion is happening in those. But for the left side of the body, it's happening in your major organs, and on the right side, it's happening in the lungs. So one pump is to the lungs, one pump is to the rest of the body. That's why it's a double pump. And Lauren Bailey says, is the pacemaker in the right atrium? Correct. Those are the natural pacemaker cells of the body. You do need to know a little bit about heart problems. So if, for example, your body, your pacemaker cells, they're not working right, your heart's beating irregularly or stops at some point, then you might get an artificial pacemaker, which is just going to work in the same way, but mechanically it's going to send an electrical signal across your heart telling it to contract, and that is what squeezes the blood over the body or through the body. You also need to know a little bit about faulty valves. So obviously your heart's working really, really hard every day, and sometimes people's heart valves, which are generally between each chamber, you'll have some valves here, you'll have some valves here. Valves, they prevent the backflow of blood. You've got them in veins. You don't have them in arteries because arteries have a really high pressure because of that small lumen, which is just the hole of the artery, and they've got a thick muscular wall to squeeze the blood through to really high pressure. Veins, they have a much wider lumen, which again is that hole, and thinner walls. They're not actually muscular walls. Your skeletal muscle is what squeezes your veins to get the blood back up to the heart. So valves, they work hard. If your heart valves become faulty or they leak, it means the heart's not working efficiently, and a person may actually become breathless and eventually die, because if their heart's not working efficiently, then the cells of the body aren't going to be getting oxygen, which means they're not going to be respiring to release energy, and so your body will just eventually die. So to do that, to fix this, they can do two things. One, they can replace them with artificial valves, or they can replace them with biological valves. So biological valves have come often from pigs or cows. Artificial valves will be made of, I'm not sure, probably some form of plastic or metal. There's good and bad about both. The good thing about artificial valves is that they're very durable. They can last for a long, long time. The bad thing about those artificial valves is that they're more likely to cause a blood clot. When something's not biological in the body, it just doesn't work as well. So you're more likely to get a blood clot, which means you'll probably have to take drugs called anticoagulants, which just means it prevents blood clots. So you'll be on those drugs for as long as those artificial valves are in your body. For the natural valves, the good thing is that you don't need those drugs, and there's not going to be as much risk of a blood clot. However, they do need replacing more often. They last about 10 to 12 years. It's the same information for a heart transplant. So you can actually get an artificial heart, but there's a much higher risk of having a blood clot, which would be fatal if it happened in the heart. So that's a big disadvantage, but the advantage is it keeps you alive for long enough to actually get a biological heart transplant. Okay. To answer Laura's question, do we need to know where they put an artificial pacemaker, or is it just where the pacemaker cells are? You don't need to know where they put the artificial pacemaker, apart from in the heart or on the heart, because that's where it works. You only need to know the right atrium is your natural body's pacemaker cells. Erin has said, can we go through what glucagon and glycogen are and where they're stored, etc.? That comes up in paper 2. So we'll save that for a paper 2 live stream a little bit closer to the time. If you want to find out, you can, of course, check out my videos. I have finished all of paper 2, which is great. So I've got a video on that. Yeah, so check it out if you want to. Right, Zishan, I can see that you've asked this a couple of times, so let's come back to it. How do we find limiting factors on a graph? So I'm guessing you're talking about photosynthesis. So it's good to discuss that. I'm going to use my little notebook again. First of all, in the comments, can you see if you know which factors affect photosynthesis? Have a go at writing them down. There are 3 that you need to know. Good. Joe has said light. Could you put another word after that? Yeah, I can see we've got temperature, spot on, and carbon dioxide. Again, we could put another word after that. So yeah, good, Joe. Light intensity and carbon dioxide concentration. Okay, so those are the 3 factors that affect photosynthesis. You have to be able to identify limiting factors on a graph. So we'll start with light intensity, because that's what most people said first. You put your variable that you record up the y-axis, which is your dependent variable. So we'll put light intensity up here. Oh, you know what? I just said the wrong thing. Let me flip that over. Rate of photosynthesis is what we're recording, and light intensity is what's changing. Okay, so obviously, as light intensity increases, the rate of photosynthesis is going to increase as well. But there's a certain point where that will level off, and it's where it levels off is where the limiting factor has occurred. So as soon as the line changes from a straight line to a curved line and then flattens off, that is your limiting factor. So what we would do is trace back from your straight line, and whatever value that is where your other limiting factors have come into play. So because we've got light intensity on our graph, the other limiting factors are either carbon dioxide concentration or temperature. You don't know which one, it might even be both, unless you're given additional information in the question. You just know that one of these 2 or both are limiting the rate of photosynthesis. Otherwise, it would continue to go up and up and up. Does that make sense for that one? It's going to be the exact same if we flip it around and do carbon dioxide instead of light intensity. It's going to be the exact same graph. We'll flip it and show you. CO2 concentration. and rate of photosynthesis. The temperature graph is the one that's going to be different. So again, your graph will look like this. And if you just trace across to your y-axis and read that value off, that's where your limiting factor has been reached. If you get a question, and it's quite possible you could, on limiting factors of photosynthesis, and it suggests that you use sketch graphs, then 100% do it, because you can get 4 or 5 marks using sketch graphs. And it just saves you having to write loads and loads of sentences. So I really recommend you practice drawing these and you know what they each look like. Okay, the last one is temperature. Now, temperature is different. And that's because temperature affects enzymes. Now, there's a whole topic on enzymes, and this is a really nice place that the examiners like to do some overlap between topics, which are your higher grade questions. So you might have to explain this graph. So to start with, as temperature increases, the rate of photosynthesis increases up until a point. The initial increase is because, as this is linking to your chemistry as well, as you increase temperature, it increases the rate of chemical reactions. So your reactants are carbon dioxide and glucose. You don't need to go into, what's it called in chemistry? Particle? No, there's a different word. You don't need to go into that chemistry detail. You just need to say that the rate of chemical reactions increases as temperature increases. So that's the first part of your graph. The second part of the graph, you can see, we see a sharp decrease. So have a go at commenting in the chat. Why does a really high temperature actually cause a decrease in the rate of photosynthesis? What's happening there? I already mentioned a hint. I said the word enzymes. So see if you can piece it together. Why is there this part of the graph? Yay! Lots of good answers. Well done, Erin, Lara, Hania. Sorry if I'm not saying that right. Zishan and Lauren. Everybody got it. The enzymes are denatured, which therefore means that they cannot speed up the rate of reaction of photosynthesis anymore. Okay, so that's that covered. Let's see if we've got any more questions coming up here. Hanaya, sorry, you did ask what topic does glucagon and glycogen come under. That's in control of glucose, which is topic 5, I think, homeostasis and response. So that's topic 5, paper 2 of biology or combined science, depending on what you're studying. It'll come up close to diabetes as well. Okay, some other questions. Let's see. Zishan has said enzymes in the digestive system. Oh, perhaps that was in response to Hanaya's question, which it wouldn't quite be. But anyway, okay. Can we go over what the stores of glucose are in the body and in plants? So by stores, do you mean uses of glucose? Okay, so I've got a mnemonic that I use. It's not actually a mnemonic, that's the wrong word. It's phosrac, F-O-S-R-A-A-C. Now let me see if I can remember it. So phosrac. So it's going to be fats and oils, starch, respiration, amino acids, and C, I'm going to double check. I think that's for cellulose, which is in plants. Let me double check. I think it's been a while actually since I looked at that. Let's go to my handy specification. Look up the word starch. I've got 11 results to work through. Let's see. Oh, here we go. Uses of glucose from photosynthesis. So this is just for plants. Okay, so we've got respiration, starch for storage, fat or oil, cellulose, and amino acids. Yeah. So these are all of the things that glucose is used for in plants. So phosphoric is my way to remember it. I hope that helps. Maybe it'll help you. I didn't come up with a better mnemonic. I am going to make a video on good mnemonics at some point, but maybe this isn't my best. Okay. Oh, and just to mention that in humans, we don't store our glucose as starch. That's only in plants. We store our glucose as glycogen. But that comes up in paper too, so don't sweat that right now. Okay. What is the definition of meiosis and mitosis? So, Kay Hughes, meiosis is only on paper too, but we'll cover it anyway. So, mitosis is the division, it is cell division for growth and repair. That's the function of it. While we're on it, and because I didn't mention it earlier when we talked about mitosis, I will just mention that it happens in adult, mature cells purely for growth and repair. So if you, for example, get injured, you have a cut on your skin, you're going to need new skin cells to replace that. So mitosis will happen to make new skin cells. But it will happen to make cells that are already specialized. So what I mean to say is humans are very different to plants. Plants continue to have the ability to differentiate their cells into many different types throughout the whole lifespan of a plant. Humans have their cells specialized at a very early stage, which is in the embryo. So mitosis, it really only happens for growth and repair, getting a little bit larger, you need to make new cells, repairing your cells. But for plants, they can, regrow entire limbs like trunks, etc., which goes into the stem cell topic quite a bit, but it's worth mentioning. And it happens in plants, particularly in the meristem, which is where the stem cells of a plant are. So those will be dividing very rapidly to make new cells for the shoot. And it also happens in the roots, in the meristem tissue. Okay. Oh, and you wanted to know meiosis as well. So meiosis happens in the gametes, which are sex cells. It happens to produce gametes. So it happens in the reproductive organs, the testes and the ovaries, and it is to make gametes, which are the sex cells. And they will therefore have half as many chromosomes as the parent cells because they fuse together to restore the full chromosome number, which is 46 in humans or 23 pairs. Okay, Hanaya says, can you go over plant tissues? Absolutely. So we've got different layers of plant tissues. You need to know them particularly in the leaf. So we'll start with that. So at the top of the leaf, we've got what's called the epidermis. The epidermis is a layer of thin cells, and they can secrete a waxy substance, which is otherwise known as the waxy cuticle. And this layer of cells is for protection. Got a fly, sorry. So that's the epidermis. Then beneath that, you've got the mesophyll. And I remember M for mesophyll as middle. So that's the middle layer. It's below that epidermis. We've got the mesophyll. And the mesophyll itself has got two cell types. So the first is the palisade mesophyll. And here's another little letter linking one. Palisade mesophyll, that's P for palisade, is also P for photosynthesis. So the palisade mesophyll carries out photosynthesis. And because of that, these cells are packed full of chloroplasts, we know that chloroplasts are where photosynthesis happens, so it needs to have loads of those. They're also nicely stacked beside each other, just to maximize how many there are, to maximize photosynthesis. Then so that's the palisade mesophyll. The layer beneath that is a spongy mesophyll. If you think of a sponge like you use to do washing up or showering, then it's got loads of holes in it. And the spongy mesophyll is just the same. These cells aren't tightly packed. They've got lots of gaps in between them. And that is to allow gas exchange to take place. So for photosynthesis, you need carbon dioxide to come into the cell, and then the cell will produce oxygen during photosynthesis, which will need to leave the cell. So we've got lots of like air pockets around the cells to allow that gas exchange to happen. And then the final layer of cells is at the bottom. That's the lower epidermis. And then within the lower epidermis, you've got those guard cells, which is what we mentioned at the start of the live stream. Those are what open and close the stomata. And the stomata are just the holes that let the gases get in and out of the leaves of the plant. So I think I've covered all of those plant tissues in the leaf. We've already mentioned the xylem and the phloem, which are other plant tissues. And then, of course, we've got the roots. The roots are for osmosis, which is water entering the plant. And they're also for active transport, which is to bring mineral ions into the plant. Okay, let's have a look. Endothermic and exothermic reaction, Chameleon Run Vlog says. Could you expand on that a little bit? What is it you'd like to know about that? That's maybe running into chemistry a little bit. Let me just check my spec. I think it is mentioned briefly in the respiration topic. And photosynthesis. Yep, okay, so all you need to know is that respiration is exothermic. Remember, exothermic means that energy is given out, like an exit is coming out. So respiration releases the energy. Photosynthesis is then described as endothermic, and N kind of sounds like in, so the energy is coming in. And in the case of photosynthesis, the type of energy is light. Okay, just while I mention that, make sure that when you write your photosynthesis equation, that you don't include light alongside carbon dioxide and water with another plus. That is just for the chemicals. If you're going to write light anywhere in your equation, you need to do it over the arrow. Just a little side note to remember. Okay, do we need to know the chemical equation for respiration in double a word? No, you don't. You only need to know it for photosynthesis. It's kind of silly because, I mean, it's just the reverse, like I've said before. Respiration is just photosynthesis flipped. I'll write it down for you for photosynthesis. CO2 plus H2O arrow, C6H12O6 plus O2. So that is your equation for photosynthesis. You then should be able to balance it. You don't need to do any fancy balancing here. I like balancing equations, but you don't need to know it for this one. You just need to plug 6s in front of every chemical that doesn't already have a 6 somewhere in it, which is basically everything except glucose. So you just put a big 6 in front of everything, like that. Okay, so that's your chemical equation for photosynthesis. You don't need to know it for respiration. If you did for aerobic respiration, it would literally just be everything flipped around. Okay, let's see what else we have got. What is the cell cycle like? What do we need to know about it? Okay, it's mentioned really kind of briefly in the specification and in the textbook itself. So all you need to know is that there are three parts to the cell cycle, although the specification doesn't explicitly say that. So the first part is simply that the genetic material, which is the DNA or the chromosomes, is doubled. Okay, that's the first part, doubling genetic material. As well as that, your organelles will need to be made. So you'll need to increase the number of mitochondria, the number of ribosomes, the number of chloroplasts, if it's a plant cell. Because if you're about to make 2 new cells, it's important you have enough of the stuff to go around. Okay, so first stage, creating new organelles and doubling genetic material. The second stage is then just dividing your genetic material. So the chromosomes split into the 2 ends of the cell. And the third stage is literally that the cell just splits in half. So those are the 3 stages. I don't think you need to describe them as stage 1, stage 2, stage 3. You just need to know that all of that stuff happens. So doubling of genetic material, new organelles being made, genetic material dividing, and then cell dividing. So that's all you need to know for the cell cycle. Okay, can I go over the meanings of the 3 types of variable? Yes, of course, that's really important to know. It comes up in pretty much every required practical. So your 3 variables are independent, dependent, and control variables. So the independent variable is the one thing that you change to see the effect of on something else. So for example, in your photosynthesis practical, where you change the light intensity is your independent variable. It's one thing that you're changing. And you'll probably have done this in school by moving a desk lamp different distances away from some pondweed in a beaker of water with a boiling tube inside it. So that's your independent variable, the thing that changes. The thing that you record is your dependent variable. So it's what you're actually going to write down in your results table. So in that practical, you would record the rate of photosynthesis, probably by counting the number of bubbles produced in, say, 1 minute. So dependent is what you record. And finally, your control variables, those are what you need to keep the same, because otherwise they could affect your results. So that's important. You don't just say, I would use the same equipment. I would, use the same boiling tube. That's not specific, because that wouldn't actually affect your results. It might not matter if you had a different boiling tube. What matters in this practical, for example, is that the temperature stays the same, because as we saw, temperature is something that affects the rate of photosynthesis. If your investigation is about the effect of light intensity in photosynthesis, you don't want temperature to be affecting it. So in that practical, you would need to control temperature. You could do it in a water bath. So hopefully you did do this when you did that practical. Your boiling tube of pondweed, I'm drawing a little picture, should have been in another beaker. That beaker is called a water bath, and it is going to absorb the light. Not the light. It's going to absorb the heat that's coming from the lamp, but because it's just water, the light is still going to pass through and get to the pondweed. So you should use a water bath to control temperature. So be specific with your control variables. Think about what could be affecting the results. I suggest, if you have time, look through the required practicals and try and identify your independent, dependent, and control variables for each practical. Okay, can we go through exchange surfaces? Yes, so probably want to start with the lungs for that. We've kind of gone through leaf tissue, so I'm not sure we need to go through that anymore. So we'll go through lungs, and then if I've missed thinking of any others, let me know. So the lungs are where gas exchange happens, and there are some adaptations for this. So you've got your lungs obviously branch off into a network, into your bronchi, your bronchioles, and then your alveoli. The alveoli are where the gas exchange actually happens, and they have some adaptations. So first of all, they have a very thin wall. That is going to make it easier for the gases to get in and out. They have an excellent blood supply, so there are lots of capillaries surrounding alveoli, and they are very well ventilated. So that means that lots of air is coming in and out of the alveoli, maintaining that high concentration gradient for gas exchange to take place along. So that's the alveoli, and that's gas exchange in them. Can you do monoclonal antibodies? Sure. So this, I think, is higher tier only. I've just seen Varisha ask if we're doing a specific tier. Varisha, I'm covering everything. I will try and point out if something is combined or triple, so that you know for what you're studying. So I'll just check. I think monoclonal is either higher or biology only. Okay, it's both. So it's biology only and it's higher tier on biology only. So I'll keep it brief, as I know not everybody is studying that. So monoclonal antibodies are made when a mouse is injected with normally an antigen or the whole bacteria. So when you inject the mouse with that bacteria, its body and its immune system is going to respond, and it will respond by making the correct antibodies for that antigen. Remember, the antigen is just a protein on the surface of the bacteria. So if this is your bacteria, then this is an antigen on its surface here. Antibodies have a specific shape to that antigen so that they can lock onto it and cause the destruction of it, like this. So the mouse is making these antibodies, and we are going to take that property from it. So what happens is you extract these antibodies from the mouse cell. Sorry, not the antibodies themselves. You extract the lymphocytes. Lymphocytes are the white blood cells that produce the antibodies. And then you are going to want to make loads of these antibodies. So you take a particular cell that is a tumor cell. Tumors, they're made during cancer because it's uncontrolled cell division. And so you take the lymphocyte and you combine it with the tumor cells, and it makes something called a hybridoma. And hybridoma is special because it's got two properties. One, it can make that specific antibody for that antigen or that bacteria. And two, it can divide really rapidly, just like a tumor cell, which means it's going to make loads and loads of this antibody. So that's how it's made: an injection into a mouse, extraction of the lymphocytes that produce that antibody, and then you're going to fuse them to make a hybridoma with the tumor cells. Okay, another question: what equations do we need to know? Well, for biology, there aren't a lot of equations that you need to know. You need to know the microscopy equation. So very simply, I don't know if you like to use triangles or not. Your physics teachers probably try to make you not use triangles because they do have their shortcomings. But if you use a triangle, it is simply I am, like that. So the I is for the image, or like the drawing. The A is for the actual or the real size, and the magnification is M. So magnification is image over the actual size. When you get these magnification questions, they can be a little bit sneaky and require you to do some conversions, most commonly from centimeters or millimeters into micrometers. So I'll just quickly recap conversions because it's something that tends to catch a lot of students out. So 1 centimeter is 10 millimeters. That's really easy to remember because hopefully you'll have brought your ruler with you to your exam, and you can just check 1 centimeter is 10 millimeters. Then if you take one of your millimeters, how many micrometers does it make? See if you can say that in the comments. How many micrometers or micrometers? There's two ways to pronounce it. How many does it make? Do we know? Yeah, well done, Zishan. Makes 1,000. So if you need to turn millimeters into micrometers, you just multiply by 1,000. And if you need to go the other way around, you just divide by 1,000. So it's really important that you make sure you've got your units the right way around before you do any calculations with them. Apple, I'm afraid this is for AQA, but so much of it overlaps with Edexcel that you could listen. It's up to you. Okay, so that's one of the first equations that you need to know for your biology exam. There's one more. It's only if you're higher tier, but it's on both combined science and biology only. It's a bit of a funny one. It didn't come up last year. I'm not really sure exactly how they're going to bring it up, but it's light intensity and the inverse square rule. So what you need to know is that light intensity is inversely proportional to distance squared. Okay, so this is what it looks like. Now, I think what is more important than memorizing this is knowing what it means. So let's say, for example, we have got that required practical setup of your beaker of pondweed being a certain distance away from the lamp. The question is more likely going to say, if the distance is doubled, what is the effect on light intensity? So what you would do here, if the distance is doubled, you would put a 2 down in place of the distance, and it would be like 1 over 2 squared, which gives you 1 over 4. So, I mean, you might instinctively have thought if you double the distance, then you're going to halve the light intensity because it's got twice as far to go. But actually, the light intensity will be a quarter as strong. So what that means is just it's not a linear relationship. Linear is when things work together in steps. If you drew it on a graph, it would be a straight line. This isn't going to be a straight line. This would be a curve. So that's what it means. I don't know if it'll come up. It's higher tier only. So yeah, if you're studying higher tier, maybe take a quick look at it. Okay, Beth, you've asked, can we go through the xylem and phloem? We have already mentioned that. I can recap it very briefly. The xylem transports water. It does this, water and mineral ions. It does this by using hollow cells. In mature xylem cells, the cells are dead. So they're hollow, and they're strengthened by lignin, Lignin, and it transports water and mineral ions up from the roots. Then the phloem is responsible for transporting sugars, which are your glucose that's being made in the leaves during photosynthesis. The phloem's adaptations are that it's very long cells, or they're elongated, and that they've got little pores, which are just holes, separating the cells. And those holes allow the cell sap, or the glucose, the sugar, to move through the cells easily. So that's the xylem and the phloem. Oh, and the movement of sugars is called translocation, and the movement of water, when it evaporates through the leaves, it's called transpiration. The movement of the water all the way through the plant is called the transpiration stream. And I think that's it. I don't think there's anything extra you need to know for xylem and phloem. Okay, Zishan has said exchange surfaces, please. So I already covered the lungs. What else would you like to know about it, Zishan? Because we've also mentioned the leaf. I'm not sure what else there is. Exchange surfaces. Yeah, I think we've already covered it. Okay, so maybe the small intestines, we could cover a little bit more in diffusion. Okay. Oh, okay. Done something funny there. Okay. So the intestines have got villi. Villi are these folds inside them that look like fingers, like this. And on the surface of each villi, there are also microvilli, which are more tiny projections. The huge benefit of this is that they're going to increase the surface area. So the small intestines are the site of absorption in the digestive system. So that's where the nutrients are actually going to get into the body and into the bloodstream. So you want to get as much of those nutrients out of your food as you can. So you need a large surface area to do that, because the food is constantly getting pushed through your digestive system. So it's kind of like a race to get as much out of it as you can before it leaves the body. So I've done a quick diagram. Each one of these is a villus, which is singular, or together they are the villi. But then each tiny projection is microvilli, or microvillus, and then plural microvilli. So I'm just writing that down. Like so. Hopefully that makes sense. So that's what you need to know about the digestive system. What else? Are there any other exchange surfaces you want to know about? Zisham, let me know in the comments if you can think of anything else. Okay, somebody has asked, please, can we do digestion? Yeah, okay, so I started a little bit about digestion. So the huge part of digestion that you need to know about is enzymes. So it's important that you know the 3 types of enzymes that are working in digestion, and where they're produced and what they actually do, and where they act. Okay, so our 3 enzymes are amylase, protease, and lipase. So we'll start with amylase. So amylase is produced in 2 places. It's produced in the salivary glands, which you could be asked to label on a diagram. They just kind of look like little, I don't know, like a grape sort of shape. Salivary glands in the mouth, and then the pancreas, which is that sort of leaf-looking like organ, kind of sits below the stomach. And it's important to actually know where it sits. To me, it helps make sense of why it's produced there. So the pancreas is below the stomach and above the small intestines. It's kind of between them. Okay, so that's where amylase is produced. And amylase breaks down carbohydrates into sugar or glucose. So where it works then is in the mouth. The salivary gland is like beside the mouth, but then it actually works in the mouth. And then it works also in the small intestines. Remember I said the pancreas was above the small intestines? Well, it will release the amylase into the small intestine to work on the carbohydrates. Okay, the next one is protease. This one sounds a lot like the food that it breaks down. Is protein. So protease breaks down protein into amino acids. So it is produced in 3 places. It's the only one that's produced in 3. So it's also produced in the pancreas. In fact, all 3 enzymes are produced in the pancreas. So that's good to know. It's produced in the stomach, and it's the only one that's produced in the stomach. And it's also produced in the small intestine itself. So that's, important. It's different to amylase. It's actually made in the small intestine. Where it works then is in the stomach, because it's produced there, so that makes sense. And it also continues to work in the small intestine. Okay, so that's protease. And finally, we've got lipase. So lipase, it breaks down lipids into fatty acids and glycerol. Yeah, and that enzyme is produced in the pancreas as well. Like I said, all 3 of them are produced in the pancreas, and it's produced in the small intestines. And then it only works in the small intestine. So it's not high enough up to be in the stomach. Remember, the pancreas, food never goes in the pancreas. So none of them actually work in the pancreas. If it's produced in the pancreas, it means it will work in the small intestine. So all three of them also work in the small intestine. So that's lipase. Let's just see, I see a few comments about, a few comments about this. It happens in small intestine. Yeah, good. Ahmed, you already knew it. That's great. Okay, and thank you for responding and saying that I've already gone over the heart. Yes, I have. Mixendell, you can skip back a bit because I did a little diagram, so hopefully that will help you. Okay, what next? Please, can you go over how osmosis happens in the roots of a plant? You've answered it for me. It's the net movement of water particles from a region of high water potential to low water potential through a partially permeable membrane. That's really nice. For GCSE, you often don't have the words water potential. That actually comes up a lot more at A level. You can say from a region of a high concentration of water to a low concentration of water, rather than talking about water potential. Would be better. Yeah. Okay, can you please go over active transport? And then we want some stem cells. Okay, so active transport is pretty much the opposite. So active transport is whenever energy is required to move something against a concentration gradient. And the big example that you need to know are mineral ions in the roots of plants. So soil will often have quite a compared to the plant, it will have a low concentration of mineral ions. And so we're having to bring it against a concentration gradient. So it requires energy, and that energy comes from the aerobic respiration in the mitochondria of cells. I mean, that's kind of it. You're going to get data questions with this, which will be what tries to confuse you. You'll be given some numbers telling you maybe the concentration of ions in soil and ions in plants, and you'll have to work out what process is taking place, whether it's diffusion or whether it's active transport. So be prepared for that, a data question. But as long as you know that it's against a concentration gradient, which means it's taken from a low concentration into a region of a higher concentration, then that's it. That's what active transport is. Ahmed, I'm loving this. We've got 2 teachers today. So stem cells. There are 2 types of stem cells, embryonic and adult stem cells. Embryonic is taken from embryos, correct? And these stem cells can change into many types of cells. That's right, because they're going to make up the entire body. So because of that, they're actually way more useful than adult stem cells. Adult stem cells are much more restricted, and they can often only make the type of tissue in which they're found. The exception maybe being adult bone marrow. Bone marrow stem cells can differentiate into both blood and many other types of cells. But embryonic stem cells, they're the ones with the widest potential. So there is a whole section on therapeutic cloning. So therapeutic cloning, this is definitely still in the research stages. And what it is making, sounds futuristic, making a clone of yourself and then extracting the embryonic stem cells from it. So what it would be is making an embryo that is you, or it comes from the same genetic material as you, so it will contain the same genes. And because of this, there's no risk of rejection when you use the stem cells from it. The suggestions of why this would be done is in order to treat 2 disorders. And this is on the specifications, so you need to know it. And those are, let me remember, paralysis and diabetes. Because in paralysis, your nerve cells have been damaged. So if you could take stem cells, differentiate them into nerve cells, and then put them back into your body, you can replace the nerve cells that aren't working. And in diabetes, for type 1 diabetes at least, you have got cells of the pancreas that aren't producing insulin like they should be. So if you could take embryonic stem cells, turn them into pancreatic cells that do make insulin, and then put them into your pancreas, then you could cure your type 1 diabetes. So those are the two treatments that you need to know about that therapeutic cloning could work for. And yeah, that's that. Ahmed, GM crops. Yeah, that's on paper 2. Yeah, thank you. Can you get stem cells from bone marrow? Yes, you can. I just mentioned that you can get them from adult bone marrow, and they're used to differentiate into blood and other types of cells. Let's have a look at what else. You were never taught stem cells? No. Oh dear. Well, don't worry. You have got some time. I would suggest going on the specification and just reading it to start with, and then hopefully you've got some revision resources. But that was a little bit of a recap just now. Okay. Why do plant leaves have chloroplasts? So chloroplasts are able to absorb light that comes normally from the sun, but any source of light. And they then use photosynthesis to turn the light into sugars. We've also mentioned that you need the carbon dioxide and water for that. Maximilian, is this Edic cell? No, this is AQA. Like I've said, a lot of it overlaps, so you absolutely can tune in anyway if you're studying Edic cell, but I'm a specialist in AQA. Oh yeah, because it contains chlorophyll, that's a nice one. Could you go over palisade cells? I already have, but very briefly, that's the photosynthetic layer of a plant leaf. It lies below the upper epidermis and above the spongy mesophyll, so it's like in the middle of those two. And the palisade mesophyll contains lots and lots of chloroplasts in those cells because that's where photosynthesis happens. That's all you need to know about that. Okay, eukaryotic and prokaryotic cells, please. Great. Okay, so these are your two big groups of cells that you need to know. When you study paper 2, you'll see that they come under the domain level of classification, but they come up in paper 1 briefly. So eukaryotic cells are cells that contain a nucleus. Prokaryotic cells do not contain a nucleus. Their genetic material is just in one loop, and it's not enclosed in a nucleus in any sort of way. Eukaryotic cells includes both animal cells, plant cells, and fungi. Prokaryotic cells can include protists, bacterial cells, basically. Yeah, right, eukaryotic is animals. Don't forget that it's also plants as well. Okay, so that's the big difference between them. You then can you need to know about the eukaryotic cells in detail. So the differences between an animal cell and a plant cell, and also the differences in bacterial cells. So animal cells, they've got mitochondria, ribosomes. Do we all know what ribosomes are for? Have a go at commenting in the chat what ribosomes' functions are. There is a bit of a lag. I apologize for that. Yeah, well done, Kay Hughes and Laura. It's for protein synthesis. Okay, so they've got mitochondria, ribosomes, a nucleus, cell membrane, which is what controls what can enter and leave the cell, and they've got the cytoplasm. So those are the 5 organelles in an animal cell. Let's go on to the next eukaryotic cell. We've got plant cells, and they contain a few extra things. So they've got the cell membrane, but outside of that, they've also got a cell wall. So if you're asked to label it, make sure you don't mix those two up. The cell wall is always the very outside layer. The cell membrane is the inside one. So look really carefully at where the arrow is pointing to. They've also got chloroplasts in most plant cells, but remember the roots don't have any because they're underground and there would be absolutely no need for them. So chloroplasts obviously contain chlorophyll for photosynthesis. And then finally, many plant cells will have a permanent vacuole, and that just stores sap. which is kind of like a food source for the plant. So it will keep this plant cell nice and turgid as well. So you need to know those differences. Bacteria cells can then also be have an extra layer called a slime capsule or a slime layer, and they may have a tail or a flagellum that comes out, but they might not. They might not have either of those things. The big thing is that they've got a single loop of DNA that's not enclosed in a nucleus, and they can, but again, don't always have, plasmids, which are just small circular loops of extra DNA. Jacob, what I've just said is double award. I'm covering both combined science and triple or biology only, but I will try and mention it whenever it is just triple, so that you know, depending what you're studying. Okay. Have we got any more questions? Somebody's asked for the quadrat practical. Yeah, that's in paper 2. So I will have a live stream for paper 2 at a later point. So I'll do a little bit then about quadrats. I've also got videos for paper 2 ready to go. So once you've got paper 1 over you can check out my YouTube channel. I've covered everything on paper 2 with individual videos that have got questions and answers at the end. So do take a look at that once you start your paper 2 revision. And I'm going to churn out a maths in biology video. I have started it. I'm sorry it's not ready for paper 1, but it'll be ready for paper 2. Okay, what else would we like to go over? What is relevant for combined foundation science? A lot. Basically the entire specification. I would suggest mix and del. If you haven't yet started revising, or you're not sure exactly what's paper 1 and what's paper 2, just go on to Google, type in AQA GCSE Combined Science specification, and then it will immediately come up with a button that you can download when you click on the first result. Download the specification, and then you have to know everything apart from where it has brackets higher tier. That's it. Okay, someone has said, not asked, go over the kingdoms. That's on paper 2. So, oh, there you go, por favor. Kingdoms is on paper 2. So again, I'm going to leave that. You can check out my videos if you want to do that, but I'd suggest doing paper 1 for now. Could you go over anaerobic and aerobic respiration? I've done that a little bit earlier in the video. Very briefly, we'll go through the equations. Aerobic respiration is oxygen plus glucose. Arrow, carbon dioxide plus water. It's just the reverse of photosynthesis. Anaerobic respiration in humans is the absence of oxygen. So your equation for that will just be glucose, arrow, lactic acid. And lactic acid, when it builds up in the muscles, it stops them contracting and working effectively. Muscle contraction, that's something you'll need to know in this topic or mention if you get a question like that. And then finally, your third respiration equation is in yeast. So anaerobic respiration in yeast is, again, still just glucose going in, arrow, ethanol, which is a type of alcohol, plus carbon dioxide. They make a small amount of CO2 as well. Okay. Kai Lauren Maglus says, does DNA, mRNA, or proteins come up? No, that will be in paper 2, and I think it's biology only paper 2 that you need to know that level of detail. Not actually sure, do you even need to know the expression mRNA at GCSE? I think it might just be A level. mRNA. Oh yeah, you do. Okay, well that's higher tier only. So yeah, save that for paper 2. Okay. Somebody says, guys, just cheat. Do not cheat. That's a way to lose your whole GCSE grade. You'll get nothing, I'm afraid. Okay, have we got any other relevant questions? If not, I'm just going to take a little flick through the specification and perhaps suggest a section that some people tend to avoid. I know for sure there are some sections that people don't like learning, or they just gloss over them a bit. Antibiotics we haven't talked about much. Vaccination. Diseases. Diseases is quite a chunky little section. This is one that is kind of just complete recall. So we've got 4 types of disease-causing groups. There are the viruses, bacteria, fungi, and protists. And you need to know examples of diseases, the symptoms, and in some cases, how they spread. So let's make this a little bit more interactive. See if you can tell me any of the 3 viral diseases that you need to know in the comments. So what viral diseases do you need to know? All right, I might have to delete some of these questions a little bit about sex. So not that it's not biology, it's just not quite relevant for this chat. Very good, okay. Laura has identified one of our viral diseases, tobacco mosaic virus. That's really clear. You can also call it TMV. HIV, well done, Aussie, that is another one. And our third one, has anybody got it? It's not salmonella, but that does come up in a different group, K Hughes, so you do need to know that. So the third viral disease is measles. Yay, somebody just got it right before me. So Imo Grace, well done. Yes, it's measles. Okay, so those are your 3 viral diseases you need to know for the AQA specification. You do need to know the symptoms of them. So let's start with measles. Measles will give you a fever, which just means you feel very hot, and you'll get a red skin rash. It's a serious illness, and it can actually be fatal. And that, again, that's in the specification, so you need to know that. But most young children have been vaccinated against it. You might have seen it in the news recently. It's been quite topical, recent breakouts, but yeah, largely most people, thank goodness, are vaccinated. So the way it is spread is through droplets, which means like little droplets in the air, water vapor, that will have come from sneezes and coughs. So it's being expelled from your lungs containing the virus. Okay, HIV then, it's one you've probably heard about. It's often discussed when we talk about safe sex. Here I am mentioning it. So this is a flu-like illness, and it can lead to AIDS, which is like the late stage of the HIV infection. So it's really bad because essentially when you have AIDS, it means that your immune system isn't functioning properly anymore, and you're unable to fight off really common illnesses like the common cold. So it's really damaging for your immune system. So it is transferred by sexual contact or exchange of bodily fluids, for example, with needles that drug users share. Okay, the third one that was mentioned was tobacco mosaic virus, or TMV. So this affects the tobacco plants or other plants, including tomatoes. So what happens here is it gives a mosaic pattern. I don't know if you've ever seen mosaic tiles, but those are like those tiny little tiles. Sometimes you might see them in a swimming pool or something, and they're colored, and it's got like a sort of spotting pattern. So this one and rose black spot fungus, those are both really likely to be linked to the photosynthesis topic. Both of these can affect photosynthesis. So TMV will discolor the leaves, and when they're discolored, that means they're not green anymore, which means there's a lack of photosynthesis because it's lost its chlorophyll, and so the plant is going to suffer. It's not going to be making as much glucose. Since I've mentioned it, rose black spot is the other plant pathogen that you need to know about. This one is a fungal disease, okay? And it presents with sort of bluey, blacky, purpley spots on the plant. Again, these spots are going to mean reduced photosynthesis, and actually the whole leaf can turn yellow and drop off. That level of detail is in the specification, so make sure you do learn that. Of course, if the leaves have dropped off, they're not going to be photosynthesizing, which is bad news for the plant. And this disease, it can spread by water or wind, so it can spread pretty rapidly. But it can be removed or treated using fungicides and just getting the plant and snipping off the affected leaves. So there are some ways to treat it. Okay, can you name me any of the 2 bacterial diseases that you need to know and give me the symptoms or how you would know if a person has it? So the 2 bacterial diseases. Let's see who can come up with those and the symptoms. Okay, Kay Hughes, you got the first, Salmonella, that's correct. Ralliate, cholera, and TB, those aren't on the AQA specification, which is what I'm focusing on at the moment. But I mean, they are bacterial diseases, that's true, but you don't need them for this exam. And Laura, you've got the second one, is gonorrhea. Okay, so those are our 2, Salmonella and gonorrhea. Ozzy, that's really good. You've started to go into more detail on the symptoms. You said discharge for gonorrhea. We need to know actually more than that, and it's pretty graphic. It's not, it's not a nice thing to learn, but it's important that you know about it. So gonorrhea is an STD, and its symptoms is a thick yellow or green discharge from the vagina or penis. So that's the first symptom, that thick discharge of that lovely color. And then the second one is pain when you urinate or pee. So it hurts to do that. So those are the 2 big symptoms of gonorrhea. For salmonella, some people have said vomiting. That's absolutely right. Alongside that, you may get cramp, you may get fever, which is when you're really hot, and you'll also have diarrhea, which is when your feces or your poo is like a very, very watery consistency. So those are all symptoms caused by the salmonella bacteria. That was really good, well done. It's an STD, yeah, it is an STD, that's right. Okay, I'm going to be finishing this in the next 10 minutes. I've already overrun by half an hour of what I was planning to do. So last 10 minutes for any questions, please fire them ahead in the comments section and I'll do my best. If I don't see any popping up, I will just pick another little section that I think hasn't been discussed and that maybe we could look at. Okay, somebody said, how can you treat salmonella? Once you've got it, it's a form of food poisoning. Essentially, it will just have to pass through your system. This isn't on the specification, so you don't need to know it. But obviously, you should keep hydrated since diarrhea is happening, so you're losing a lot of fluids, and you need to replace your salts that have been lost as well. Laura says, could you find a Kahoot so then we can do a test sort of thing? I could see about that. I don't know, Kahoot's pretty hit and miss. Some of them are absolute junk. Some of them are pretty good and have been put together by teachers or diligent students. I would maybe suggest using the specification or something and asking somebody to just sit with you and test you on it is a better form of revision. No, he says, I've never been taught diseases in double award higher. I'm thinking you've got a few missing sections. So no, for you, I would really suggest that you download that specification. Like I've said before, just go on Google, AQA, GCSE, combined science or biology, whatever you're studying. I think you said combined science. And search that up. The first result will have a link that lets you download a PDF. Download that and just spend the rest of the evening reading through it and checking what you've missed. Okay, Ozzy, I have covered the digestive enzymes, protease, and carbohydrates earlier. I've gone through where each enzyme is made, what it does, and where it works. So flick back a bit and you should find that. Okay. Osmosis, active transport, diffusion. Is plant leaf structure in paper 1? Yes, it is. Osmosis, active transport, and diffusion. I think I've covered all of them. I haven't discussed diffusion explicitly. Diffusion is simply the net movement of substances. Either it can be liquids or gases. Both have particles which can move. So it's the net movement of particles from an area of their high concentration to an area of their low concentration. So common examples you need to know for this exam are diffusion of gases in the lungs and diffusion of gases in the leaf tissues. Can you briefly go over the required practicals needed? Yes, and differences between bacteria, virus, fungi, and protists. Yes, okay. Oh, loads of questions coming now. Okay, but I think most people are looking for the practicals. Okay, so this is going to depend on what you're studying. Can you, maybe in the comments, can you comment what course you're doing? I want to know if we've got any triple science students or if we're all combined science. So tell me in the comments, what are you studying? Combined Science Foundation, Combined Science Higher, Biology Foundation, or Biology Higher? Let me know. Okay, we have got a triple student. We've got 1, 2, 3, 4, 5. Oh, maybe that's triple for dark shadow. Okay, we've got a mix. Okay, so I'm going to cover both then. All right, so required practicals. Again, these are on your specification. I don't have time to go through each and every required practical, but I think there are other resources that you could hopefully find on YouTube to help you with your required practicals. So, required practical 1 is use a light microscope to observe, draw, and label a selection of plant and animal cells. So, obviously, you're not going to be doing that in person in your exam. What you are going to be asked is to use the microscope equation, the magnification equation, to work out any one of the 3 variables on that. I mentioned this a little bit earlier in the live stream, but this is the equation in a triangle. So you've got the magnification, the image size, and the actual size. So that's your equation. If you're not sure how these triangles work, this line here means divide, and when the letters are beside each other, it means multiply. Then you just cover up what you need to know. Okay, so let's say you're asked to work out the magnification, and you're given the image size and the actual size of, say, a ribosome. All you do is cover up the M, and you would get the image size and divide it by the actual size. It's really important that these are both given in the same units. So let's say one of them was given in millimeters and the other one was given in micrometers or micrometers, same thing. You would need to convert them. So here's the conversion. To turn millimeters into micrometers, you're just going to multiply by 1,000. I'll write that on there. Going this way, you times by 1,000. And going back the way, you divide by 1,000. Like so. So just make sure that you check the units. They do this a lot in physics, but it can happen in biology too, where they try and trick you out with the units. That's largely it. You might, for example, have to label an animal or plant cell, but you'll have revised that within paper 1 anyway. Okay, sorry, I dropped my pen. Okay, the next required practical is a biology only required practical. So you only need this one if you're doing triple. This is investigating the effect of antiseptics or antibiotics on bacterial growth using agar plates and measuring zones of inhibition. Okay, there's quite a lot of health and safety and processes in this practical. You need to know, for example, that you need to sterilize both the Petri dish and the culture medium, which is basically what the bacteria grow on, so that you've got a pure culture of your bacteria. You also need to sterilize your inoculating loop, which is the metal loop that you use to spread the bacteria on the agar jelly. And then you need to place your antiseptics on it. Again, those need to be disinfected each time. So you'll have like a beaker of disinfectant. You place your antiseptic discs onto the culture, and then you're going to leave them. So you'll place the lid on the Petri dish. You'll use some adhesive tape or sellotape to seal the dish. It's important that you don't seal the dish the whole way around. If you do that, you are stopping any oxygen getting in and creating anaerobic conditions, and more dangerous pathogens or dangerous bacteria, which are pathogens, they tend to survive more in anaerobic conditions. So you want to make sure that you let oxygen get in. So you'll just seal it on the sides or in an X shape over the Petri dish. And then you will flip it over so that the agar jelly is on the top, so that no condensation from the lid drops onto the agar jelly, which could affect your results. Then you'll incubate it. Do you know what temperature you should incubate at and why? Have a go at answering in the chat box if you think you do. Yep, well done, Laura. You've got that spot on. 25 degrees Celsius is what you incubate at. If you incubate it at a warmer temperature, for example, 37, that's too close to human body temperature, and you might grow dangerous pathogens for human health. So you only ever incubate at 25 in a school laboratory. Okay, that's how you do it. Then you need to analyze your results. Zones of inhibition are basically where there is no bacterial growth any longer. The bacteria kind of looks cloudy on your Petri dish, kind of bright. So where you've got your antiseptic disc, there will be like a clear zone around it. See this little zone? That would be your zone of inhibition where no bacteria are growing anymore. And you're simply going to use your circle pi r squared formula to work out the area of that. Okay, so it's just pi r squared. You need a scientific calculator. Please make sure you all bring one to your exams. Okay, that's that one. Then the next required practical is investigate the effect of a range of concentrations of salt or sugar solutions on the mass of plant tissue. Okay, so this is when you've got different concentrations and you want to see the process of osmosis happening. So water will either enter or leave the plant tissue depending on the range of the range of concentrations in there. This really depends on you knowing your definition of osmosis and just applying it. Okay, so it's quite a long one to explain. Let me draw a diagram. If the sugar, we'll just talk about sugar solution, but it's going to be the exact same for salt solution. If the sugar solution is a higher concentration of sugar in the water than in the, we'll call it potato chip, it could be a carrot, it could be a whatever plant tissue. If it's a high concentration and it's higher than in the potato, then water is going to move out of the potato. And when it moves out of the potato, it loses mass. So you would weigh all of your potatoes at the start. It's important that they have been patted dry every time you weigh them, because any excess water or solution on your potato chip will affect its mass. So that's just a little part about how to carry out the practical properly. Okay, so water will move out if there's a higher concentration of sugar in the solution than there is in the potato. If it's the other way around and you have got actually a very low concentration of sugar solution, like 0.1 moles, then what will actually happen is that the water will probably move into the potato. And in this case, the potato will gain mass. So when you weigh it, it will have got heavier after a few hours. Okay, what you're going to then get, sorry, I think you can't see it. What you're probably going to be asked to do is draw a graph of this information or interpret a graph of this information. It's quite a data heavy required practical. And you'll see that you've got some potatoes gaining mass and some potatoes losing mass. So as the concentration goes up, the potato will lose mass. So let me draw a little tiny graph and hopefully I can explain it and it will make sense for you. Concentration of sugar would go on the bottom and your percentage of mass gained or lost would go up the side. Okay, so when the concentration of sugar is really low, let me just check. Then it will go into the potato, so it will gain mass, and when it's really high, it will lose mass. So it will look like this. Okay, don't let this graph scare you. The question that you're likely to get with this is, what concentration is the potato's sugar? So basically, if the potato and the sugar solution are the same, then no mass is lost. So all you have to do is go to where the mass lost is 0, which is this middle line, read across, and whatever value this is of the concentration of sugar solution, that is going to be also the concentration of sugar solution in the potato chip. Okay, so this practical, it can be given to you in quite a hard way with graph questions, or the potato osmosis practical can be given to you in a really simple way, like identify some equipment that you need for this, just to do a calculation or something like that. I don't know. It could be an easy one. It could be a hard one. Okay, the next required practical is the food test. I've already covered all of that earlier in this pod or in this live stream, so please just go back. That was quite near the start, so flick back for those. Number 5 is investigate the effect of pH on the rate of amylase enzyme. Okay, so in this experiment, you will have got amylase and some starch. When amylase breaks down starch, it turns it into sugar. From the food test practical, hopefully you know that iodine is used to test for starch. And iodine is blue-black if starch is present, or yellow-red if starch isn't present. So, yeah, I can save this, don't worry, it's no problem. Let me get some paper. The way this practical is probably going to be done, probably how you've done it if you've done it at school, is in a spotting tile. You'll have a mixture of both amylase and sugar solution in each one of these little tiles. And every 30 seconds, you're going to move across and you're going to add iodine to each of the spotting tiles. And if starch is present, it goes blue-black. If it's been broken down, then it will just stay orangey red. What you're looking to see is how long does it take for it to stop turning blue-black. So this would be 30 seconds, 60 seconds, 90 seconds, 120 seconds, and at 150 seconds, that's when all of the starch has been broken down. Okay, so that's first of all the general principle. Then we need to apply it in terms of enzymes. So you will do this whole experiment for each different, I just said enzymes, for pH. You're going to do this experiment for each different pH. So maybe this is pH 5. And at pH 5, it takes 120 seconds before that starch is broken down. You make the different pHs just by adding a solution of a different pH to your amylase and starch or sugar solution. Okay, so what you should see is that amylase works better at higher pHs. And if you recall your digestion topic, that's because amylase works in, as well as the mouth, it works in the small intestines, and the small intestines are alkaline. Take a look at bile if you want to know about why they're alkaline and how that works. Okay, so this is the amylase and effect of pH on amylase. You could be asked to draw a graph with this as well. Let me know if that makes sense. Tyra says, okay, you know how last year's paper had carrots, not potato, would it be the same formula? Yeah, any plant tissue. They could literally choose anything, a pumpkin, butternut squash, anything. They'll all follow the same principle. Okay, next required practical is required practical 6. We're getting there. Is the light intensity on the rate of photosynthesis using an aquatic organism such as pondweed. I've already briefly mentioned this one. Essentially, you're just going to have a light source, which is probably going to be a desk lamp. For this one, by the way, if you get like a 6-mark question, draw a diagram. It's going to make your life so much easier. So you have your desk lamp. You're going to have probably a meter ruler, and you're going to be placing your beaker of water, which is your water bath, with the boiling tube of pondweed at different distances from the light source. So it's going to look like this. This is your setup. What you're going to do is you're going to record the number of bubbles that are produced in a set period of time. I would suggest you say 1 minute. That is long enough to actually count the bubbles. So you're going to count how many bubbles are produced in 1 minute when your pondweed is 10 centimeters away from your light source. What you're then going to do is you're going to move your pondweed along the meter ruler. So maybe you move it to 20 centimeters away. Now, do you think it's a good idea to immediately start counting the bubbles for the next 1 minute? No, it is a bad idea because the pondweed hasn't had time to adjust to the new intensity of light because it's further away. So you need to give it, and I think it's recommended 5 minutes, 5 minutes to adjust to the new light intensity. So each time you move the beaker of your boiling tube of pondweed, you need to let it rest for 5 minutes. Okay, and that is going to give you a range of results. So you'll see how many bubbles are produced in 1 minute, and that tells you your rate. Let me think. Oh yeah, I did mention this before. The water bath is important because it is going to control the temperature. So that is your control variable. Okay, let's go on to the next practical, required practical 7. Plan and carry out an investigation into the effect of a factor on human reaction time. Okay, so because they've said a factor, this means you could be given a lot of different scenarios in your exam. For example, you could be given the effect of caffeine on your reaction time, the effect of amount of sleep, whether you're tired or not, on reaction time. It could be the effect of practice on reaction time. Do you improve the more that you practice? It could be pretty much anything you can think of, the effect of chocolate on reaction time, the effect of drugs on reaction time. So there's not one thing exactly to revise for it. But what you do need to know is a general suggestion of how to measure reaction time. The most common one is that you have got a meter ruler. This is my meter ruler, it's just my pen. And you will hold it above the hands with the 0 in line with where the hand is. And your partner who's holding the ruler is going to drop it, but they're not going to tell you when they're going to drop it. And what the person does is they try and catch it. Not like that. Where they catch it is where you record the distance of how far the ruler has moved, for example, 15 centimeters. And that is then used alongside a conversion table to work out the reaction time in seconds. Because remember, time it's not measured in centimeters, it's measured in seconds, milliseconds, etc. So you need a conversion table to work out the reaction time in that practical. Then you will be exposed to whatever factor has been chosen. Maybe if it's caffeine, you do it before having a coffee, and then you do it again after having a coffee, and you will compare the reaction time. Here, it's very important to have repeats to make sure that you get enough results. You don't just do it once each time. So do repeats. And yeah, that's most of it. Just make sure you make it really clear that there are no other signals to tell the person when this is about to be dropped. It has to be completely random. If the person counts down 1, 2, 3, drop, that's not testing reaction time because the person is expecting it. So it has to be dropped without warning. Reaction time is paper 2. Oh, have I gone on to paper 2? You are absolutely right. I have. Sorry, that's it. So we actually finished there with the photosynthesis equation. Who was that pointed that out? Ahmed, my second teacher here. Thank you for pointing that out. Yeah, okay, forget about reaction time until you start revising paper 2. Okay, that's it. That's all of the required practicals covered. This has been 2 hours long, which is longer than I planned. I really, really hope that you all do really well in your exams, not just tomorrow, but for the whole exam period, and that you have some extra confidence from this. If you did enjoy it, please subscribe. I would love you to watch some more of my videos, and please give me some feedback, some thumbs up, some comments, ways to improve. I'll be doing another live stream for paper 2. This is my first ever live stream, so I'm really happy that so many of you joined. It's really kind to see all your nice comments thanking me. That's really sweet. Yeah, I'm here to help you. So thanks for coming and helping me. All right, thank you guys. I'm on Instagram as well, so you can check me out there too. Bye.